So hopefully part of my talk will actually address that last question as well. So the title of my talk is Maximizing Accuracy of Cellular Deconvolution. So what really spurred a lot of this research is exactly that question, is how do you trust the annotations from these single cell reference sets, and how do you optimize them for downstream deconvolution? So really, this is really the overarching theme is, the pursuit of precision medicine, right? How do we better understand the tumor microenvironments within, in my focus, cancer cells, to really make sense of the different cellular components that are within those cellular niches. And so we provided our package to hopefully address some of these problems. Right. So the human genome came out about 25 years ago. That's about when they started working on it, and it came with a great promise that we would be able to understand human genetics, leading to better disease outcomes, right? The traditional model of medicine, where we're treating, a group of patients, all with, say, colon cancer, with a single therapeutic, hopefully treating everybody and everybody gets better. But in reality, some people respond and some people don't respond. And the worst case scenario, some people actually have adverse effects. So by using some of these newer techniques like single cell sequencing and now spatial sequencing, right? If we can start developing better precision targets for these patients, can we actually lead to better outcomes, tailoring therapy to those individuals? Unfortunately, in the past 10 or so years, we really haven't seen a tremendous outcome in the clinic. So things haven't really improved yet, and it's because of the challenges within that tumor microenvironment. So I'm mainly focused on head and neck cancers, and it's a really interesting cohort because it's across 6 different anatomical locations, right? So we have multiple etiologies, so smoking and tobacco use, as well as infection with HPV, and they cause different changes within that cellular environment based on the source of that cancer type. There's a really high rate of recurrence despite therapeutic intervention and surgery. Nearly 50% of all patients will have recurrence, leading to eventual treatment resistance, recurrence, and metastases. And this is the heart of the problem, right? All these tumors are very, very heterogeneous. There's a mixture of different cells. Recent research has shown that different parts of the tumor have completely different profiles than other parts as well. So using some of these techniques, can we better address these types of problems? A recent paper even demonstrated that depending on the immune profile within these tumors, there's actually hot and cold tumors that will respond or not respond based on the actual complexion of those tumors of the immune cell population in there. So what we thought is, okay, if we can use these deconvolution analysis using all the new reference sets, the new single cell reference sets that are available, can we start really understanding the tumor microenvironment dynamics, hopefully improving treatments? So these are some of the questions that we're trying to address. Really, the top one is, how do you actually know if a reference set, and when I say reference set, I'm talking about a single cell reference set, so a tumor that's been profiled using single cell or spatial technology. How do we know if it's suitable for deconvolution? What are the technical biases in the upstream processing? So there's a lot of eloquent solutions for the deconvolutional process, as we just saw one. But upstream of that, how do we optimize these references for use as a reference set going into this? And then can we develop a robust strategies to recommend or not recommend certain data sets to be used? So we just saw a really good overview of how deconvolution works. So really quickly, tumors are profiled using typically single cell RNA sequencing or bulk RNA sequencing. Not a lot of them have done both. There's very few data sets out there that have done both on the same tumor, but there are a few existing for head and neck cancers, which is very, very helpful for us. So you profile both of these samples using bulk and single cell. You run it through your algorithm of choice, whether CyberSort, Music, there's a dozen or so now. And the end goal is to get that readout, that cellular proportion readout. The advantage of reference-based deconvolution, which is going to be the focus of this talk, is you have those labels, right? It makes interpreting that data set when it comes out way easier than if it was label-free. However, there's many, many challenges with the reference-based approaches. It's really sensitive to reference quality, and it's dictated by the quality of those resolutions and annotation choices upstream, right? If someone took a look at this UMAP here and said, oh, okay, well, those fibroblasts, there's 3 separate clusters there. How do you know that they should be grouped up like that? Should they be split out? Those are the types of decisions that have to be made before you enter into the deconvolution process. The resolution of data set matters as well. So these are three of the more popular head and neck single cell reference data sets out there. And you can see from the 2017 data set, it was kind of really broad labels. And then as we kind of proceeded, we got more and more and more specific as, you know, better annotation, better markers, as people got a little bit more comfortable with the technology. But the more granular resolution, the more finite labels aren't necessarily a good thing for a deconvolution process. So how do we know which level to look at? There has been a lot of algorithms benchmarking performance of these different tools, but none of them really have focused on the upstream processing. So like, how do you choose the clustering parameter, for example, the resolution of that single cell data set? They focus on, you know, how does transformation of the data set matter for deconvolution? How do transformations affect it? How does the actual algorithm that you're running affect the outcome of deconvolution? But really upstream, not a lot of work has been done to look at that. And to illustrate this problem, this is the same data set with 8 different resolutions, right? It's very, very difficult to just kind of look at the data and say, okay, I think resolution 0.1 is the optimal solution for this, right? The default, the more popular tool, Seurat, is 0.8. In this particular data set, that default parameter would be way, way over clustered in my opinion. So we took kind of a step back as we were working through these data sets and said, you know, can we develop a tool that just simplifies this process and gives us a little bit more confidence before we enter into deconvolution, right? Can we devise some sort of metric or some sort of system to say, okay, yeah, 0.2 is a good solution for this problem. So we developed an R package to do this. And this is kind of the general workflow of deconvolution. And right there in the middle is our ACeD optimization, as we call it. And the goal of this is to really aid researchers in choosing that optimal reference set. We start with an input reference, and this can be a publicly available one or one that you generate in your lab. You do pseudo-bulk it to start. You take all the samples that were sequenced for that particular sample, create a pseudo-bulk profile of it, and then use that as a query to that reference-based deconvolution. So you use that as like your bulk. While this isn't the perfect solution, it's because it's very difficult to get gold standards for all the different cell types within these tumor populations. So it's right now the best we have. But once you do that, you get a readout of all the different proportions for all the different resolutions for all the different samples. What we started looking at was the different metrics that can be used. So we started with the root mean squared error. And what we saw was that as you increase the resolution, essentially your root mean squared error drops continuously down. So theoretically, the best solution would be to continue to add more clusters. And it's because the number of cells assigned to those clusters get down to 1 or 2. So you get singleton clusters. So we needed a better metric to identify, you know, the optimal solution. So we came out with a metric called ACeD, and this is weighted based on the total number of cells per that cluster. So this curve here is running the same single cell data set as reference across all these different resolutions. And we basically try to pick out that top point on that plot of where would be optimal. You can see there's a kind of a neighborhood of points that provide a pretty good solution. And I think it's up to the analyst to kind of look at that data set and say, okay, does this make sense based on the marker genes, based on the clustering result as well. There is a relationship between the resolution and the final cluster count as well. So in the yellow color there is a more optimal solution, or what we call more optimal. And you can see there's actually plateaus. So a bunch of different resolution parameters will lead to the same clustering output, but actually give slightly different scores. So within a given clustering, there might be a more optimal solution for that deconvolution run. So in this case, the optimal resolution for this particular data set was 0.31. And we just kind of do, at the moment, a relatively simple brute force approach where we give it a range of different clusters, step through all the different resolutions, and see what comes through. So hopefully you can make it a little bit more sophisticated in the future. But you can see the output here on the top to bottom. The optimal solution in this particular case is 10 different groups, right? So if you look in that red at the top, those were the original fibroblasts. A more optimal solution is, in fact, to split those 3 fibroblast populations out into 3 distinct cell types. But it doesn't always prefer to split things. You can see the CD4 and the CD8 T cells actually was a more preferred solution was to keep them together. So in this particular case, we weren't able to separate out those two populations based on this particular reference data set. So as a proof of principle, we wanted to see, okay, when you apply it to real data sets, in this case, this is the TCGA head and neck data set, how well do we actually constitute some of the known group structures? So the little red lines there on the y-axis of these plots are HPV status. So after deconvoluting using the default resolution, we actually lose that HPV signature based on cellular proportions. Whereas a more optimal deconvolution using 0.31 of that reference set, we get a lot more clustering of that HPV group on the right-hand side of that plot. The other really important and interesting thing to note is that optimal resolution is reference set specific, but it's also specific to the algorithm that you're using. Some algorithms might perform better with a specific reference. So I tested two algorithms. I've tested a few algorithms, but for this purpose, gedit3 and music, which are two of the popular ones. You can see they actually came with slightly different outputs, right? gedit3 produced a 10 cluster resolution, and the music algorithm came out with 8. So not hugely different, but there are differences, right? The epithelials were split out at the bottom for gedit3 where it was grouped up for music. We also extended this to developmental system as well. And this was a really interesting test because of that problem of like continuous development. So this is a mouse, embryonic mouse retinal system at embryonic day 13.5. So there's a progenitor cell population that gives rise to 3 distinct cell types. There's a lot of overlapping gene expression throughout those progenitors into that transitionary state in that center there. So that center would be in like that C3, C4 group. And you can see ATOH7, for example, really starts in that transitionary state, but by the time it gets to the end stages, it goes down in expression. Conversely, OTX2, which is a photoreceptor-specific marker, continues to go up throughout that development, but it starts in that transitionary state. So it's tricky for deconvolution because there's no discrete separation between these different cell types because it is a trajectory model, like a cellular fate model. So how would a deconvolution algorithm perform is really kind of up in the air. It just depends on where those lines are being cut. So we ran it through the ACeD algorithm to figure out what would be optimal, and it actually preferred to group up those transitionary cells into one bigger group because it wasn't able to specify between the select few markers that were actually differentially expressed between those different groups. We do see an interesting kind of ladder down through the scores. So that suggested to me that there was only so many finite solutions for this particular problem, but it also grouped up the progenitors there as well. The other interesting thing about this data set is we actually had a knockout system as well. So we knocked out that ATOH7 gene, which essentially removes the one population down in the bottom there in that cluster 2. And the optimal solution, even though we kept the transitionary states to be kind of a single group, we still perform quite well. So the heat map there is the actual proportion in the single cell compared to the estimated proportions in the bulk samples that we had sequenced. So this is one of those rare data sets that we actually did single cell sequencing and bulk sequencing on the same samples as well. So it gave us a really nice test case to use. So currently it's developed as an R package and it's integrated with the Seurat workflow. We're still in the testing and development phases. We would like to add a bunch more deconvolution algorithms so the user has a choice of what they're using. And we also would like to kind of do some benchmarking and see which tools perform better or perform worse in certain situations. So if you have a data set that you're interested in trying it on, let me know. I'd be happy to collaborate and work through it with you. But yeah, and we'd also like to do a little bit more of a sophisticated strategy as opposed to just walking through all the different resolutions. For some of the faster tools for deconvolution, it's no problem, but some of the tools can be a little bit slow. So if you're doing 20, 30 different resolutions for each object, it takes a little bit of time. So, and then with that, it's a great group of people at UB that we're working with on these projects. So yeah, take any questions.
Questions? If nobody has a question, then I ask. So regarding, maybe I misunderstood the setup, but so you have single cell RNA data on which you're essentially optimizing with essentially your training part in there to optimize the clustering. So did you look at sort of generalizability across other data sets? Because it seems to me that you possibly now may overfitting quite a bit towards algorithm and the specific use case.
Yeah, and absolutely. Yeah, so in this particular case, we trained it on a single cell model, then applied it to 500 tumors within the TCGA, and it performed pretty well. So we're trying to optimize a single reference set. So I agree, like, because we're training it on its own pseudobulk profile, you're optimizing the deconvolution on itself. It's a little bit circular. Ideally, we would have other standards that we could use to train off of. And there are some, but not necessarily for like solid tumors.
Cool, thank you. Any other questions?
Hi, I have a quick question. Thank you for the wonderful talk. So I'm wondering, when you choosing the reference for your cell deconvolution, do you have to consider the patient sample status, like whether they are healthy or whether they are coming from different subtypes of the disease, whether they are in more advanced stage or in the very early stage? If that's the case, how do you deal with that, whether it would affect the efficacy or accuracy? Thank you.
Yeah, that's a really good question and something we're actively working on. So luckily in head and neck, we have about 15 pretty decent single cell data sets now. Within the last 2 years, there's been a huge increase in data. So within just these 3 data sets, though, there's dramatic differences in deconvolution outputs. And I believe it's because of that hidden cell type problem, right? So that first data set that was generated in 2017, which was like a foundational data set for head and neck, you can see it's pretty sparse and we're actually missing cell types. We're also looking at atlases. So basically combining all these different head and neck sets together, generating like a cell atlas, and then you're running deconvolution on it. So we have a much more representative of the population. And I think that's probably the better way to go. But it's just a question of data availability. So.
Okay.